So I want to talk about two things, a brief introduction to the field. I'm sure all the individual speakers will give a little bit more specific information, but I just want to give a little bit of history as to how we've got to this technique. And you'll see a lot of what I mentioned in my talk repeated, but I think for certainly those of you who've never used gene editing or are new to the field, one or two of these slides could be quite useful. So where do we start? A bit of an anathema to physiologists, at least in times gone by, but I think in recent years, the last couple of decades or so, there's been a real effort to bridge this split which occurred back in the 1950s between calcium signaling and ion channels in cells and Watson and Crick's DNA molecule. But that's a really good place to start if you want to understand gene editing. So this is the famous 7'6" structure in the middle here, which really doesn't tell you very much, but this is a much more informative diagram from the original paper drawn by James Watson's wife. And what it shows is the anti-parallel nature of DNA, which people often forget about the double helix. We've got one strand going up, and we've got one strand coming down. And this is the final sentence from this paper, which I think is really worth having a look at. It's not escaped our notice that the specific pairing mechanism that we've postulated, and I'll explain that on the next slide, immediately suggests, maybe if your brain is the size of a planet, but there's no lack of confidence in these guys, a possible copying mechanism for genetic material. So let me explain what they mean. We'll just turn this molecule on its side, and we'll do it in some pretty colours. This is the chemistry that they're on about. They've got base pairs, cytosine and guanine in yellow and red, and we've got adenine and thymine. So this is the base pairing. The DNA, as I said, it's anti-parallel strands. You've got one going in this direction, one going in this direction. And as you know, DNA is labelled 5' to 3', and this is all down to the chemistry or the names on the different parts of the carbon molecules and the sugars. And the mechanism that they're on about is this. This was the structure they proposed, and they said simply if you pull the two molecules apart, then you've got all the information to make two new strands. Just by having short DNA primers which match in a base pair specific manner, you can then start to fill in the gaps just by following these rules. Red base pairs with yellow, green base pairs with blue. And if you follow this through, you can get one complete molecule as shown here, and you can get another one. Oh dear, what's happened? This is what's known as DNA damage. This happens fairly regularly that you get a double-stranded break in a molecule. But this is okay during the process of DNA replication, because this damaged chromatid here can use the information stored in this one, as shown here. We can have a resection event, which allows the two molecules to interact, and the chromosome at the bottom can copy the details from the one at the top, bridge the gap, and then go back to its day job, carry on synthesizing DNA, and just fill in the gap, and DNA repair has occurred. So this process, known as homologous recombination, is happening in all our cells every day as we're speaking at the moment. But this is critical to understanding gene editing, as I'll show a little bit later. What about mutations? Mutations cause disease. This is a slide from 1957. Ingram's work identifying the mutations which cause sickle cell anemia. And as you can see from this slide, if you look carefully, there's just a single amino acid difference. And if you look at the sequence here, you can see it's red, green, red. At the bottom, it's red, blue, red. So it's just one change in the genome which causes this disease and 7,000 different other genetic disorders. I suppose the ultimate goal for many people of gene editing is just to change that sequence back, get the normal sequence, and successfully treat disease. You'll hear a lot more about these options this afternoon. So what is editing? Well, this is the first editing experiment by Mario Capecchi back in 1982. And this is a very similar scheme to the one I've shown before. Here we've got the genome in blue. We've got an individual gene, which is a neomycin resistance gene. But this red bar signifies a stop codon in the gene, which stops it from producing an active protein. Second part of the system is a donor molecule, a sequence which mimics the chromatid which you've just seen in the previous slides, which has the correct sequence at this region, but also has a stop codon, which means that this molecule by itself can't express the functional protein, neither can this one, but if the two of them interact, you can get neomycin-resistant cells. So this process works, but it's incredibly inefficient. That said, if you take these cells, put them into a mouse blastocyst and go through a breeding program, you can win a Nobel Prize. What you've also done is revolutionised the study of both genetics and physiology by the process of making what we call knockout mice or gene-targeted mice. And for many, many years, this was the only real way to study this, and it really was limited to the mouse system. But an experiment performed by the same group, round about the same time, 1984, was this simple experiment in bacteria, or with bacterial plasmids, I should say, in mammalian cells. What we've got here is just a key to explain these lines here, which are just linear representations of circular plasmid molecules. This plasmid contains the full-length neomycin gene, and if you put this plasmid into cells, you get a large number of clones grow up which are resistant to neomycin. A very simple experiment they did was then just to truncate this neomycin gene slightly. It's called DR, deletion rights. If you put this in, nothing happens. If you make a similar clone, deletion left, put that in, you get no colonies. They then did this experiment and put the two plasmids in together, so neither coding sequence by itself can work, but they can recombine, and you get a small amount of recombination. Now, if you've just dozed off, wake up again, because this is the really key part to the whole of the symposium. They did a really simple experiment. To one of these plasmids, just before they put it into cells, they did that. They made a targeted double-stranded break in the region here, and one of these molecules being broken before they put it into cells allows it to interact much more efficiently with the other molecule and get a very large number of colonies. And a second important experiment they did was to make another double-stranded break, but much further away. And having this double-stranded break much further away massively reduces the efficiency of this process. So the key to gene editing is being able to make double-stranded breaks close to the region that you want to. But in 1984, this was a problem because the only enzymes that you had were restriction enzymes, which recognise 6 base pairs. If you do the maths, 6 base pair sequences occur roughly once every 4,000 base pairs, which is fine for a plasmid, but it's absolutely useless for a genome. You cut it into three quarters of a million pieces. So there was a long wait in the field. People knew what to do, but didn't know how to do it. This was solved initially by designer nucleases. Now, designer nucleases, all of the nucleases you'll see today have a recognition sequence. Very different mechanisms, but they all recognise at least 18 base pairs. Because now if you do the maths, you have a recognition site which occurs once in every 7 times 10 base pairs, 10 to the 10 base pairs. So this on average will cut less than once per genome. So this should be a very precise cleavage event. Here's a pivotal experiment in the field from '96. This is the first zinc finger nuclease. These are artificial proteins. These proteins don't exist in nature. They're chimeric proteins which bind to DNA sequences, 9 base pairs on this side, 9 base pairs on that side, and a dimer of nucleases which can make the double-stranded break. This is lambdaphage, and all this did for this experiment was generously donate its genome of 48 kilobases. And all these zinc fingers do is cut a little bit off the end. You can probably, sorry for shouting, just about make it out there. There's a very small band at the bottom, there's this large fragment at the top. So this was the first demonstration that you could do these large-scale cleavage events. But it took another 10 years before this experiment was performed in human cells. And this paper came out in Nature. This is the IL-2 receptor gamma chain, and this shows that many genes have got lots of different mutations which can cause disease. And they tried to fix this using zinc finger nucleases. And it was this paper that made me interested in gene editing just over 10 years ago. So what did they do? Well, let me just explain this assay, because you'll see similar assays reported during the course of the day, and it might be useful trying to work your way through those. This is a bit of DNA in the genome, and these are just primers for PCR. And if you do a PCR across this region, you'll get a product of about 1.5 kilobases. They then added a donor to the sequence, and the donor does 2 things. It corrects the mutation, but it also adds a restriction site. So if this restriction site ends up in the genome, when you do your PCR and then digest the PCR product with BSRB1, if it's been successful, you'll get 2 bands, not 1. But if you just put the donor in by itself, nothing happens, or nothing that you can detect anyway. If you just put the zinc fingers in by yourself, you don't get the repair that you're looking for, but something else slightly interesting happens. You'll make a double-stranded break, and then these two ends will come together and shorten the DNA ever so slightly. And this is a process known as NHEJ, non-homologous end joining. Simply the two ends stick together, and as we now know, we lose a few base pairs on either side of the junction in many cases. But this allows us to knock out genes. That's fine, but we're after precision repair at this stage. However, if you put the two together, a double-stranded break in a genome, human genome for the first time, this now allows these molecules to interact. Copy this sequence, and if you copy this sequence, you should get a PCR product. You should get incorporation of the BSRB1 into the genome. You get a PCR product, which when you treat with the enzyme, now gives you two bands. So this shows that a proportion of the products, proportional to the intensity that you see, have got this incorporated. This is a process known as homology-directed repair. This gives us precision repair of the genome. Just a little caveat here. You'll notice these two things I've boxed here. G2 arrested, there's a drug called vinblastine they used in this experiment, which boosted the efficiency about 2.5-fold. And the reason it boosts the efficiency is because it arrests the cells in this phase of the cell cycle where homologous recombination is most prevalent, which is the S phase, the synthesis phase. So if you want to engineer cells using this technique, they have to be actively dividing. If they're not actively dividing, then this technique won't work. Non-homologous end joining works at all phases of the cell cycle, but there's a very significant reduction in the efficiency of HDR if it's not in the S phase. So this really was a pivotal paper, and this is what got our group in Cork interested in gene editing. Now, what can you do with gene editing? These aren't our experiments, but these are some of the experiments which came out then in the first few years after this. As I mentioned before, all gene editing in animals in vivo have been done with mice. I remember talking to my old head of department in Cork 10 or 15 years ago saying, you always work on rats for the kidney. Why is it that you don't actually take advantage of the transgenic systems and then match up the genetic data with the physiological data? And then he showed me a rat kidney and showed me how he cannulated it and said, you really want me to try this in a mouse? And this was a problem. We have a technology for modifying mice, but not other animals. So this is a transgenic rat. It's engineered to express GFP. And the beauty of the gene editing technology is that you can just take a fertilized egg from this animal. So if you've got any kind of in vitro fertility system set up, you can do this. You then take the zinc finger nucleases, either as a protein or as an RNA molecule encoding the proteins, or as a DNA molecule which can make an RNA molecule to make the proteins, and inject it into the pronuclei of one of these fertilized eggs. What will happen in a lot of cases is that you'll get gene knockout. You'll get these non-homologous end joining reactions occur in one gene. And if you transfer these knockout eggs into a pseudopregnant female, she'll give birth to animals, many of which are knockouts. In some cases, up to 40% of the animals will have one allele knocked out. Some of them may be chimeras, so you'll still need to go through 1 or 2 cycles of breeding, but you can very rapidly generate these knockout animals. And this is the small menagerie of animals where this has been successfully done with other gene editing techniques in the last 2 or 3 years. We've got mice, rats, cows, sheep, rabbits, even monkeys, zebrafish, and people are now starting to do this. There's one group licensed at the Crick Institute here in London to work on human embryos up to day 14. Here's another really powerful paper which was made by Kathy High's group in 2011. And this represents here part of the factor 9 gene, which is defective in haemophilia B. There's 4 exons here, and these little black lines represent mutations. I say black lines, they can occur anywhere throughout the gene. And as I just showed you with the previous example, with the interleukin 2 receptor gamma chain, you can get mutations at lots of different places. So does this mean that we need individual zinc fingers or gene nucleases and individual donors for each exon? Not at all. What Kathy High's group have done in this paper, and many other groups have done this, and you'll see examples of this in other talks today, is they created what some people call super exons. So take the wild type sequences of exons 2, 3 and 4, fuse them together, which if you've done any molecular biology you'll realise is just a truncated cDNA, and use this as a donor, and then design your nucleases to cut in one of the intron regions. This will then recombine and then the endogenous genome exon 1 can splice to the other exons, give you normal messenger RNA and normal protein. So this has several advantages. Not least, you only need one zinc finger and one donor molecule, but it also takes advantage of the endogenous control sequences, so you get much better temporal and spatial control. It makes it much more physiologically relevant than just adding cDNA into cells. Another really important thing is shown in this slide here, and that is that gene editing is, to all intents and purposes, a permanent change in the genome. This is from the same experiment from Kathy High's group, and what they did was they compared gene editing to cDNA addition. These are the liver of these mice which they've added the gene editing system to, and after 10 weeks you can see that you've got a fairly good level of factor 9 being expressed. This restores hemostasis and these animals behave normally. But if you do a partial hepectomy, cut some of the liver out, over a few weeks it grows back, and what you see is there's an initial decline, but then as the liver starts to regenerate, the levels of factor 9 are maintained, and that's because the cells which are regenerating have been permanently edited, and the new cells that they make also have this change permanently in there. If you do this with the cDNA liver though, do the hepectomy, the cells grow back, but they've lost the cDNA. This is a transient process. So it's a much more stable event. It's been used in the clinic. You'll hear more about this. This is one of the first studies doing a knockout of the CCR5 receptor. For those of you who don't know, HIV has two receptors, CD4 and a co-receptor CCR5. But there's some people who don't have CCR5 naturally and they're resistant to the virus. So this experiment used zinc finger nucleases to delete the CCR5 gene, knock it out, and in some cases you saw up to 1,000-fold reduction in viral load, and this has already been used successfully in at least one patient. But this is an interesting graph. This is the papers published per year on zinc fingers, so a little bit of a slow start from people working on it, and it is a little bit tricky to start in the first instance. But it then kind of plateaued about 3 years ago. So why this technology, which has revolutionised things, why did it kind of grind to a halt? Well, part of that reason is because talons were described in around about 2011. Talons can do all of the things that zinc fingers can do, but have at least two advantages. One is, for many cases, they're much easier to design and synthesize, and they're also more specific, and again you'll hear a couple of talks on this a bit later. They've also been used in the clinic. Many of you will have seen this on the newspapers about a young girl at Great Ormond Street Hospital who had an incurable acute lymphoma. She'd tried all the different treatments and the patient was told, or the parents were told, there's nothing else we can do. But then she was treated with these CAR T cells, that's the tumour cell expressed in CD19, and the CAR T cells, it's a chimeric antigen receptor which can bind to these cells and destroy them. The only problem is your own immune system destroys these donor T cells before they can act. So selectors developed these talons which can delete the T cell receptor on the surface of these T cells, and this allows these T cells which are added to the patient to avoid the immune system for long enough to destroy the tumour, and at least in one case this had a very, very positive outcome. But the thing that's really taken off is CRISPR. This is the number of papers here. We're already up to nearly 2,000 papers in just 3 years for CRISPR. So what is it that CRISPR is so good at? Well, I can teach you in less than a minute how to make a CRISPR, and I think this is key to the whole process. Here we've got a sequence of DNA, and I've highlighted two things here, the first of which is the PAM, or the proto-adjacent spacer motif. And it's just 3 nucleotides. In this case it's TGG, and this is part of the recognition by the protein, and it's just the 2G residues which are important. The T is simply a spacer. And what we're going to do is design an RNA molecule to bind this sequence here in the red box of 20 nucleotides. So that's the design process, we're done. We just need to follow the rules. G binds to C, T binds to A, and design this RNA molecule. We just make it in the context of a slightly longer RNA molecule called a guide RNA, and we need to express it with this yellow shape here, which is the Cas9 nuclease. So Cas9 is a nuclease, the guide RNA directs it to the right site, and we get a double-stranded break, 3 base pairs upstream of the PAM sequence. You can deliver this as RNA protein molecules. You'll see some people using this a little bit later on. But the approach we've used in the lab is just to make a plasmid to express this. So again, I'll briefly talk you through this. We just need to make if you look at the RNA sequence there in blue, we just need to make a DNA molecule equivalent to this and add an extra few nucleotides to clone it, as I'll show you in a moment. We make a second oligo, which matches this base pair specific and a 4-base overhang here. This is €10 in 2 days. And then clone it into a plasmid such as this, which contains the Cas9 protein and the core part of the guide RNA sequence, a promoter for Cas9 and a promoter for the guide RNA. The Golden Gate cloning technique is possibly the easiest cloning technique you can come across. You cut with BBS1, this flips out this little insert, the ends that you've put on match perfectly to the gap that you've created. And if this goes in, you can't cut it out again because you don't have the BBS1 sites. Once you've got this plasmid, you just stick it back into cells and this will make the protein for you. And you hear a lot more about different delivery techniques later on in some of the talks today. So what about testing these nucleases to see if they work? Can they make double-stranded breaks? Well, if you do a PCR across a region, I said before, it's difficult to tell by PCR that you've made a deletion. Many times it's only a few nucleotides or maybe a +1 insertion. If you do get a mixed population like this, where one of your PCR products is slightly smaller, if you run it on a gel, they'll all look the same. So a really simple technique was developed a few years ago, whereby you simply boil your PCR product, which denatures them. It doesn't damage them. You then allow them to mix for a few minutes so they reassort, and then cool it back down to room temperature, take it out of the kettle, and then these molecules will reanneal. But if you've got a short molecule reanneals with a long molecule, you'll get these little bubbles. And many of you will have heard of either the CEL1 assay or the T7-1E1 assay or the surveyor nuclease assay. They all work on the same principle of being able to cut these molecules where you get the bubbles form. You'll get 2 smaller fragments and you run it on a gel and it looks a little bit like this. In reality, it looks like this. And I'd just like to acknowledge Kieran Lee in the room who did this gel several years ago when we were trying to get this assay set up. One thing that people always ask me about CRISPR is, off-target effects. Well, the first off-target effect paper came out in 2013, and this slide in itself suggests that it hasn't been a major problem for the field. It's something we can't ignore, but it's not as bad as everybody first wrote out. This paper I'm talking to just showed this data here. So just very briefly, this was the target site for the CRISPR, and you can see the sequence here, but you can see the cutting frequencies in other sites which have got 18 out of 20 base pair matches. So this level of off-target cleavage is actually quite bad. But the same group, this isn't the same paper, it's the same group a year later, showed that simply truncating the guide RNAs, just making them 1 or 2 or 3 bases shorter, slightly destabilizes the molecule and then makes it much more dependent on the right interactions. And at least in this example here, this is a really good example, you detect no off-target effects. There'll always be some with certain guide RNAs, and again you'll see this discussed during the course of the talks this afternoon. There's also new variants of Cas9, enhanced specificity Cas9. So these are molecules which have had certain amino acids changed. So this lysine converted to an alanine. Lysine binds to the negative charge on DNA molecules. Removing a few of these lysine amino acids, converting them to alanines, slightly destabilizes the protein, and in many cases causes a big reduction in the off-target effects. Again, you'll see discussion of these different proteins during the course of today. So just to summarize this first part, what Cas9 guide RNA does, what talons do, what zinc finger nucleases do, what meganucleases do, all of them generate double-stranded breaks. We've then got 2 options. If we have a donor plasmid or a donor oligo, we can get homology-directed repair. These are these precision repair events which only occur in cells which are actively cycling, and the efficiency here is generally relatively low, although with selection schemes you can boost this a little bit. We've also got the default pathway, which is non-homologous end joining. This is donor independent. We can make small deletions, or you'll see later large deletions, and this is generally a much more efficient process, but this can only be used, generally speaking, to knock things out. And a message which is really important, and as many of you have found, anyone can edit with CRISPR. All you need is a lab, about €100 to buy a plasmid to get started. And where many people start is adgene.org. If you just Google adgene and CRISPR, you'll find a very good repository of information. Now, I've got about 4 or 5 minutes left, so I just want to briefly explain some of our work on CF. I won't get through it all, but I just want to make a couple of points. For normal lung function, we need this protein, the CFTR protein. It's an anion channel. It allows bicarbonate and chloride to pass through, and this sets up an osmotic gradient which allows water molecules to pass through. And this creates a nice healthy lung with cilia beating, slippery mucus on top, and any germs or dust which come in we can cough out and expectorate. But if you've got mutations in CF, then this process doesn't work and the cilia collapse, we get a thick sticky mucus, and we get this very debilitating and life-shortening disease. There's different classes of mutations. These 3 here are all very severe. This protein we have reduced conductance, this protein we have reduced trafficking, but with these mutants we don't make any protein. There's really good drugs for this one, there's licensed drugs for this one, but for people with class 1 mutations, there really isn't that much in the therapeutic pipeline. The great hope was that gene therapy, cDNA addition, would help these class 1 patients, but this is the data from the most recent trial. This was a multi-dose trial where people got CFTR cDNA every month, and if you can see, the best result is a mild stabilization of disease. Now, the people who did this still aren't fully sure why the gene therapy didn't work, but one possibility is that the cDNA went into the cells and was very rapidly lost. So our question is it time to edit? So I just want to show you 2 more sections. The first is using our approach on HDR for single mutations, and this was set up initially by Rowan Flynn in the lab and then subsequently in collaboration with Kieran Lee. So the first paper by these 2 guys was to use zinc finger nucleases, which Rowan designed, and then worked with Kieran to make the donor and see if we could repair the mutation. And the problem at the time was we couldn't get these zinc finger nucleases particularly close to the CTT. But a couple of years later, when the CRISPR system came out, we were able to redesign and move our Cas9 nuclease quite a bit closer to the CTT, which is the region which is missing. So for CF, we have a 3 base pair deletion. This is the donor, which contains these 3 missing base pairs. And this slightly complex slide at the top shows the location of this. It's about 90 base pairs upstream of the delta F deletion. And our donor, we also had several other nucleotides in here so that we could measure the extent by which the DNA repaired. Now, people often ask what CRISPR stands for. It stands for clustered, regularly interspaced, short palindromic repeats, which really tells you nothing about the mechanism. But I think this is much better. Cut, resect, invade, synthesize, precision repair. So let me show you what I mean. All that these nucleases do is cut the DNA. Cellular proteins then cause resection events, which then create these 3' ends, which can invade the donor. They invade the donor. The next thing that they do is to copy or synthesize DNA. So the chromosome now is using this as a template. This then rejoins, it then fills in the gap and synthesizes the other strand, and we end up with precision repair. I'll skip through these slides, but what this showed is that at best we got about 2% gene editing efficiency. A big improvement on our previous work, still the best that anyone's reported in CF without selection. But if you're going to use this therapeutically, what you're going to need to do is to take these cells ex vivo, select and enrich them, and then try and put them back. Now, we've got a grant at the moment to do this in collaboration with Steve and several other colleagues. But the other approach that we've tried previously is this one, and I'll finish up on this. This is using non-homologous end joining. This has several advantages. This occurs at all stages of the cell cycle. Many cells in the lung are very slow dividing cells, so maybe we need to use non-homologous end joining. It'll be very efficient, and as I'll show you some of our data, we get up to 50% knockout. But how can knockout fix a gene? Surely that doesn't make sense. Well, when we do knockout, we get end resection, the 2 ends join together, fill in the gap. We've sequenced it. Sorry, I knew that would happen. These sequences have got misaligned, but we get small deletions. This is just a dendrogram of the distribution. Most of them are only about 10 to 20 base pair deletions. we can use 2 guide RNAs and cut out a specific region of the gene. In this first test that we did here, we got nearly 8% knockout. So the way that we can use knockout is to take advantage of the fact that for CF there's at least 2,000 different mutations which cause the disease, 3 of which are shown here, are located deep within introns. These are the names, the numbers underneath refer to the patients who have these particular mutations. And just to look at one in particular, the 3849 plus 10 kb, 3849 refers to the last nucleotide of exon 22, plus 10 kb means 10 kilobases into the intron sequence here. We have a C residue, which when converted to a T, creates this pseudoexon. It interferes with the splicing. It creates a site which isn't normally there, which the splicing machinery recognises and goes, oh, there must be an exon here. This then introduces an in-frame stop codon, and we get a truncation of the protein, which gives rise to cystic fibrosis. So the question we asked very simply was, can we cut this region out using CRISPR? So we made different guide RNAs which cut on either side, and David Sands, who's in the audience, showed that we could get efficient cleavage using different combinations, in one case up to 70% of transfected cells. Does this change the splicing? Because this is what's really important. Well, the assay that David developed was shown here, making these 2 plasmids. This is this intron region and exon 23, and what we see here is normal splicing. When he changed just one residue, this C to a T, this is what we predicted would form the pseudoexon, and what you see here is a big shift in the splicing over to the mutated form. But when David co-transfected guide RNAs in Cas9, we remove this region and we shift the editing almost back to normal. I don't have time, but we've done this for 2 other mutations. Again, it's the royal we, this is all David's work. So I don't have time to go into the multiple mutation scheme here, so I'll just finish with this slide on CF gene editing. We know that HDR is precise, but it's inefficient. But if you've got a suitable ex vivo route, and you'll see people describing this today, and you can select these cells, then this is a very good therapeutic avenue. But if you're going to try go directly in vivo, then we reckon that maybe for some mutations, the non-homologous end joining approach will be better. It's very efficient in our hands, in one case up to 70% of cells, but for CF patients, it's only going to work for about 2% of people. I didn't have time to show it, but there's some data from Tony Catherman's lab using the super exons which I described initially for the factor 9 gene. Tony earlier this year, just a couple of months ago, showed that by homology-directed repair, you can use super exons to correct up to 80% of the CF-causing mutations. But again, it's very inefficient. You can select the cells, but maybe for therapeutic purposes using in vivo editing, that wouldn't work. But we and many others are now working on non-homologous end-join super exon strategies, and hopefully it won't be too long before we can report some data on that. Finally, and you'll see this referred to many times today, delivery is always going to be a challenge, whatever we do, but I think we're getting there. Okay, thanks very much.